Welcome to the 85th Cold Spring Harbor Symposium on Quantitative Biology. As you can see, we're doing this virtually this year. I am Brian Ray. I'm an editor at Science Magazine, published by the American Association for the Advancement of Science. And I've had the pleasure of attending this symposium, one of the most prestigious meetings of the year, I think. And the topic this year is biological timekeeping. And the organizers have very generously offered me the opportunity to sit down with some of the speakers and to discuss some of the results that we heard and some of the most interesting advances that are going on in this field of biological timekeeping. So with me now is Katja Lamia, who is at the Scripps Research Institute in San Diego. And Katja, before we start, may I just ask you, you trained in physics actually as an undergraduate and in biophysics. And so we're interested sort of in how people end up in this field of chronobiology. And can you just tell us a little bit about your path that led you to do research in this area?
Yes, absolutely. So yes, I did my undergraduate degree in physics. And when it came time to go to graduate school, I really wanted to do some research that was more medically relevant for personal reasons. That was interesting to me. And so I applied to biophysics programs and, not really knowing what that was going to be. I thought I might do structural biology. But then when I started, I wanted to do lab rotations and a lot of different things. And just really, didn't know much biology. So I really wanted to expose myself to different areas. And two things during my first year in graduate school actually introduced me to circadian rhythms. First was I subscribed to Science Magazine, and the very first issue that arrived at my apartment had the cover was a picture of Drosophila with green fluorescent protein under the control of the PER promoter. This was from Steve Kay's lab. I'd never heard of circadian rhythms, but I read the paper and I thought, oh my gosh, that's so cool. And partly with my physics background, I was really intrigued by this idea of having a biological oscillator. And then I did a rotation actually with Steve Reppert, who was then at Mass General Hospital. And, great experience, very exciting, learned about the field. But I had another lab rotation with Lou Cantley, and I just loved his lab. And I thought it would be Lou Cantley's lab would be a great place for me to learn all different kinds of science and be introduced to different techniques and aspects of biology. So that's where I did my PhD. But I kind of kept this interest in circadian biology, as my own kind of side interest throughout my PhD studies. And during my PhD, I ended up studying insulin signaling and metabolism. And I noticed that if we did experiments at different times of day, the outcome would be different. And so I got interested in the possibility that there might be a molecular connection between circadian rhythms and metabolism. So actually, when it came time to decide what to do for a postdoc, that was one of the things I was interested in. I also applied in labs that were focused on mass spectrometry, which again, I thought it would take advantage of my physics background, and that was really an emerging, exciting technology. And I seriously considered that too, but I ended up going with circadian biology and haven't looked back.
That's great. It's one of those areas that's more interdisciplinary, and I think becoming more important these days, that people understand things like the physics of oscillators and then bring that into biology so that we can really understand how these systems work. I think one of the important areas that got talked about a lot at the meeting and that circadian biologists think about is this idea of people's own biological clocks and what happens when those are out of sync with the daily cycles that they're experiencing of feeding and activity in the world around them. And so I think we've all or almost all experienced jet lag, and we know that acutely when you get shifted away from what you're used to and you're trying to be active, you're trying to eat in another time of day, it doesn't feel that great. But then there are people in the world who do their jobs and sometimes for long periods of time shift and do the night shift and work all night and try to sleep all day. And so this appears to have really health repercussions as well, that this cannot be the best thing to do for your health. And so that this is one of the important, I think, crossovers to medicine that we heard about at the meeting. And recently there's been some discussion about whether this predisposes people to cancer. And for some cancers, there may be evidence for this. Other people have said, well, maybe the verdict is still out on whether there's a cancer risk. And so I thought, you're an expert that's working on this. Maybe to start, you could tell us what your view is. How do these epidemiological studies tell us, and can we draw any inference from them at this point?
Yes. So first of all, I'm not an epidemiologist, but I have read a lot of those papers and paid a lot of attention to it. And in my mind, there's quite a lot of evidence to support this link. There are some studies that don't find a significant impact of shift work or circadian disruption on cancer risk, but to my mind, these are very complex things. There's a lot of heterogeneity in, how the studies define shift work. There's a lot of heterogeneity just inherent to human populations. There's a lot of heterogeneity to, cancer in general, and even within subtypes of cancer. So to my mind, the evidence, there's a pretty good amount of evidence, and on balance, to me, it clearly supports a link, but the jury is still out on the details of how this works. And I think also we really don't understand, probably there are going to be subtypes of cancer that are more susceptible to having an enhanced risk by circadian disruption. And I try to think about it like, if you were asking if smoking increases cancer risk, but you didn't know which type of cancer to look at, you might have trouble, finding an association. But if you know to look at lung cancer, it's very clear, very obvious. So there may be there's probably not going to be anything as strong of an effect as smoking on lung cancer from circadian disruption. But I do think, it's entirely possible and likely that, there may be a stronger association with certain types of cancer, certain types of risk. And also, there's a couple of studies have been done that I think take a really elegant approach, where they're looking at not so much circadian disruption from shift work, but these studies that have looked at where people live within a time zone. I mentioned this during my talk. There are two really large scale studies that I know about. One was done in 2011, the author is Boris Zenkov, that looked at populations in Russia and China. And they looked at a lot of different health outcomes and mortality in general, but they did find, excuse me, an significantly increased cancer risk for people who lived at the western edge of time zones in Russia and China. And this was kind of looking at all cancers as a whole. And then another study was done in the US. The final author, I believe, was Neil Caporazzo from the NCI. And they looked at populations in 11 or 12 different states. And again, they found the same thing, that there was an increased incidence of cancer for people who live at the western edge of a time zone. For me, this really blew my mind because,
this is moving east and west within a time zone. So the amount of light that's occurring during the day is constant, right? But you're talking about the person's individual exposure to that.
Exactly. And it would just be the timing of their light exposure, as you say. And they found a significant impact, of people living only 5 degrees of longitude separate from each other, which is only a 20-minute difference in the timing of sunrise. So to me, the fact that you can measure even a small difference from, only having a 20-minute later sunrise, having an increased risk of cancer is just amazing.
So we don't want to hurt anybody's real estate values, but it's actually quite a remarkable finding.
It is. I mean, it's a small difference, of course, but it's measurable.
Yeah.
And the other thing, actually, I think is an important point is that, to me, both of those types of evidence really support there being a link. But it's also important to realize that this doesn't necessarily mean that it's the same mechanisms at play in both cases. It could be that the timing of light exposure enhances the risk of, some type of cancer by one mechanism, and circadian disruption from shift work might actually work through a different mechanism. It might not even be the same, but, to me, on balance, both of these types of evidence suggest that there is a strong link with some aspect or maybe multiple aspects of circadian rhythms that are influencing cancer risk.
Yeah, it's a really important point that you make. We lump this word cancer on a great variety of diseases, and the characteristics of them are not certainly not shared across the spectrum. And we know a lot about that. The more we learn, I think maybe the more difficult it gets, because we do have to sort out those subtypes and how they respond. But you've also taken this further and said, okay, we can look at these, what happens to people, but maybe we should take this into the lab, take this into our own hands and do some experiments. And so you described to us that you can make mouse models that are particularly designed to use some of these genes that we know are drivers in human cancers, and then predispose those animals to cancer, and then you can control the light cycle, give them a little mouse version of jet lag or sort of like shift work, anyway, a disruption of their circadian rhythm, so that they experience that problem, and then look at how that influences cancer incidence. And so can you tell us what you're finding in those studies?
Yep. Yeah, so we like to use these mouse models partly because they're much easier to control, and we can look at a very well-defined system. And I also want to say that I did not develop these mouse models. They've been developed over decades by experts in cancer biology. In particular, the ones that we're using were first generated by Tyler Jacks and Dave Tubison. And actually, it was a paper from Tyler Jacks's lab with the first authors, Thalis Papagiannakopoulos, a few years ago, that first showed that one of their models of lung adenocarcinoma can be influenced by circadian disruption. And so it was based on that study that my lab decided to adopt that model to try to understand, what are the molecular mechanisms underlying that effect. And so we've used the models that they developed, and in our hands, excuse me, it's actually a model of lung cancer that's driven by expression of oncogenic KRAS only in a small number of lung cells. So the model, the tools that they invented and developed allow us to turn on the expression of oncogenic KRAS only in a very small number of lung cells by introducing a virus that we can introduce into the lungs at a low dose. And so only a small number of the lung cells turn on the expression of this oncogene. And that is known to be sufficient to initiate the formation of lung adenocarcinomas. And in the study published by the Jacks lab a few years ago, they focused on a model where they turned on KRAS and also deleted P53, which is a tumor suppressor that's one of the most frequently mutated deleted genes in all cancers, in particular in lung cancer. And they could see an effect of circadian disruption on that model. But in our study, we see actually a much bigger effect on the model where we only turn on oncogenic KRAS. And as I mentioned in my talk, that may just be because it's a milder way to initiate tumor formation. So the tumors take longer to develop. They have a longer exposure to chronic jet lag. Maybe that's why we see a bigger effect. Or it may be that there's really a role for P53 and the mechanisms that are underlying the disruption, which we don't know that yet. But so we were able to recapitulate and their finding, and also show that we see an even bigger effect when we're only turning on KRAS. And so that gave us a system to try to, start dissecting what's happening at the molecular level in response to chronic jet lag that could be influencing cancer risk. And we looked at some of the mechanisms that we and others have proposed in the past. So for example, our interest in this was really sparked by a previous finding from my lab where we showed that the circadian core clock component Cry2 can regulate the stability of c-MYC, which is another really common oncogene. But in this study, in this particular model of KRAS-driven lung cancer, we do not see an increase in c-MYC in the tumors that were from mice exposed to chronic jet lag. So that does not seem to be the mechanism in this particular case. So then we took an unbiased approach to look at, what's happening to gene expression. And we found, to our surprise, that so there's huge amount of heterogeneity, as you might imagine. We're looking at tumors, which are going to be heterogeneous, looking at a response to circadian disruption, which causes heterogeneity in gene expression and the timing. But we could measure a small but significant increase overall in the expression of genes that are activated by a pathway that responds to temperature increase. So there's a transcription factor called heat shock factor 1 that is responsive to temperature and other types of stress that basically put stress on the amount of protein in a cell. And the pathways that are activated by this heat shock factor 1 enable efficient protein folding, generally, but also one of the hypotheses is that this can enable the folding of mutant proteins that are required for to support tumor growth and formation. So, this was a surprise to us. We weren't expecting this at all. But then we go back and read the literature, and there's actually quite a lot of literature suggesting a connection between circadian rhythms and heat shock factor responses. Most of this was done by Uli Schibler's lab in their efforts to understand environmental signals that set the timing of circadian clocks. And there's also a couple of papers from Joe Takahashi and Ethan Brewer that support this as well. So this further, sparked our interest and, supports the idea that this really wouldn't be too surprising for this to respond to circadian disruption. And then there's also a wealth of literature suggesting that heat shock factor 1 can support tumorigenesis. And this was mostly done by Susan Lindquist's group, and now some of her former trainees are continuing that work, Mark Mandillo and Chen-Kai Dai at NIH. And so we are now getting reagents and tools from them to follow up and try to understand whether, we've made this observation that heat shock factor responses are increased in chronic jet lag, and the KRAS-induced tumor burden is increased in chronic jet lag. And there's some evidence that heat shock factor 1 could support that, but we need to test whether that's really the reason that it's happening. So we've gotten tools to inhibit, either pharmacologically inhibit heat shock factor 1 or genetically delete heat shock factor 1 and determine whether that's really required for the increase in tumors.
Yeah, so this is really promising. It sounds like the system is allowing you to drill down and to test in a particularly good unbiased way as to what the changes might be that are going on in these cells that could be contributing to the process as they respond to the jet lag. I think you also told us that in some cases you see increases in tumor number, right, but not necessarily the size of tumors. That correct? So
Yeah, right.
to me, that's both promising and daunting. It's promising that you have the ability to characterize these different properties of the tumors, but it also shows you that the effect of circadian misalignment may be on a particular part of tumorigenesis, not globally, just of affecting on or off cancer or not, but perhaps we have to understand which particular aspects of tumor formation rather than tumor growth are particularly susceptible to this kind of disruption.
Yes, I agree. Yeah.
All right. Well, thank you so much for taking the time. I think it does seem clear that we all should be paying attention to keeping ourselves in tune with our circadian rhythms so that we don't suffer these kind of effects. And the more you give us insight into how this works, the better we'll be able to protect and deal with these issues. So thanks so much for taking the time to speak with us.
Of course. Thank you for giving me the opportunity.
You're welcome.